here on the outside are each composed of alternating sugar and phosphate groups. The sugar, in fact, is deoxyribose. And these groups are linked together by strong covalent bonds. And linked to each sugar, also by a covalent bond, here and here, for example, are these flat ring-like groups, which are bases. There are actually 4 different kinds of bases, which we've shown with different colours, but I'll return to their more detailed structure later. For now, just notice that the bases on the 2 chains lie opposite to each other in pairs. And notice also that they're linked together, but not by strong covalent bonds. Instead, there's a kind of interaction which we've represented by these dashed lines, as here, to indicate that they're weak bonds. In fact, they're hydrogen bonds. There are other weak bonds in DNA which run vertically between the bases, but we haven't shown these because it would make the model too complicated. The thing to remember is that the DNA structure is stabilized by weak bonds, and this is an absolutely general feature of all the big biological molecules. Well, now let's go and look at the base structure in more detail, because the bases really are the key component of DNA. Well, we haven't shown all the detailed atoms of the bases, just their overall shape. This red one is adenine. Notice it's got 2 fused rings, and these rings are composed of carbon and nitrogen atoms, with side chains coming off. Adenine's attached to its deoxyribose at the end of this side chain here. This green base, guanine, is very similar in shape to adenine. In fact, it differs only in the nature and position of some of its side chains. Adenine and guanine belong to the same class of base. They're called purines. But now this brown base here, thymine, it's rather different. It's only got 1 ring, but again, there are side chains. And this yellow base, cytosine, very similar again to thymine, but also differing in respect of its side chains. Well, thymine and cytosine belong to another class of base called pyrimidines. So you see, we've got 4 types of base in DNA. 2 of them, adenine and guanine, are purines, and the other 2, cytosine and thymine, are pyrimidines. But remember that each base in DNA, and let's take cytosine as our example, is covalently linked to deoxyribose sugar, which in turn has a phosphate attached. This general grouping of base, sugar, phosphate is called a nucleotide. And more precisely here, because of the nature of the sugar, it's called a deoxyribose nucleotide. Now, there are some very important molecules in cells which are just single nucleotides with different bases and different sugars. The point about DNA is that its 2 chains are polymers of nucleotides, in just the same way that the chains of a protein are polymers of amino acids, and of a polysaccharide, they're polymers of sugars. All these big polymer molecules are called macromolecules, big molecules. And DNA is just one example of this. Well, now let's get back to our central problem, which is how you get to a structure like this from this. Well, the first thing you need to know is where in the cell your macromolecule is. And then, how do you get it out and purify it so that you can study it?
These are plant cells. They're stained with a purple dye that reacts specifically with DNA. It's the nucleus that is stained, therefore DNA is in the nucleus. But how to get it? Bacteria are a better starting point than plant cells for 2 reasons. They can be grown in great quantity, and their DNA is naked inside the cell. It's not surrounded by a nuclear envelope. Here are bacteria stained as before. You can see where the DNA is. Now, how to get it out of living bacterial cells? Here's a way using a homogenizer. This will show you the principle of the method. The tube contains a broth of bacteria. The piston nearly fits the tube. cells are agitated violently and smashed open. Their DNA spills out along with the rest of the cell contents. This is the sort of thing that must happen but the DNA is broken into fragments in the process of extracting it. It's been subjected to too great a force. If I want intact molecules, I've got to use a more gentle technique, like this. That's the way to do it. If suitably prepared bacterial cells are put into pure water, they swell up and burst, and their DNA just floats out. This electron micrograph shows a single bacterial cell that has been burst open gently. Surrounding the cell, there's the DNA from inside, which is now spread out. Once the DNA has been set free, it can be concentrated and purified. Here, a suitable bacterial broth, in which the cells have been opened, is added to cesium chloride. A dye is added to the mixture. This will help find the DNA later on. Now a sample of the mixture is taken up and put into a centrifuge tube. The sorting and separating of DNA is a slow process, but can be greatly speeded up. Now, here's the trick. The cesium chloride is very heavy, and the cesium ions begin to settle in the bottom of the tube. As they do so, a gradient of density is built up. The DNA will separate out and come to equilibrium at a point where its density is equal to that of its surroundings. The process, even with the centrifuge, takes about 4 days. The end of day 2 and day 3 and day 4. And now you can see that bright band. That's where the DNA is in this tube.
So now we've extracted some reasonably intact DNA from bacterial cells, and we've also shown how it can be separated from the other cell components and purified. But in fact, you can get some very useful information from the unpurified DNA. Because it's such a big molecule, DNA after suitable treatment can actually be viewed in the electron microscope. And you've already seen this electron micrograph of DNA, and now this great spaghetti-like mass of DNA spewing out of the central broken bacterial cell is actually one molecule of DNA. Well, it's obviously a very, very long molecule, and it's uniformly thin all the way along. It's 2 nanometers wide along the whole of its length. Some very patient people have also unscrambled pictures like this and worked out the lengths of DNA molecules. It turns out that for a bacterium like this, the molecule is about 1 millimeter long, and that's 1,000 times longer than the cell that it came from. So there's a unique feature of DNA. There's an awful problem folding it up and packaging it so that it fits into the prokaryote cell or the eukaryote nucleus. So then, for the really big macromolecules, we can use the electron microscope to determine gross physical characteristics, shape and dimensions. But what about the chemical nature of DNA? How do we know what it's made of? Well, a useful first step is to break it up gently down into its component bits by hydrolysis with acid.
Chemists have a test used to demonstrate the presence of deoxyribose sugars in a solution. This is a positive result. A second test can be used to demonstrate the presence of phosphate. This again is a positive result. So both components are present in DNA. In all samples of DNA, the amounts of deoxyribose sugar equals the amount of phosphate present. The acid treatment also liberates the four bases, and these can be separated using a method called chromatography. I'll demonstrate the method. First, I'll mix two inks, one blue, and I'll add red on top. Now, as a solvent flows up the paper, the pigments stream out of the ink mixture and move up the paper. The blue is quicker than the red, so the two pigments begin to separate. All the blue up at the top, the red sluggishly behind. To separate the bases, a small amount of acid-treated DNA is put onto chromatography paper. It's put on a pencil-marked line to help locate the spot later. The sample is now dried. This happens very quickly. The dry paper is now hung in a chromatography tank. It has to be handled carefully to keep it clean. The point of using the tank is to create controlled conditions under which this separation of bases takes place. The solvent is added to a tank at the top. The solvent will run along the paper, through the sample, and then slowly on down the paper. After 11 hours, the solvent has got this far. But where are the bases? They can't be seen with ordinary daylight. They have to be located using ultraviolet light. Now, in these conditions, some of the bases fluoresce and so form bright patches. This one fluorescing. But others absorb ultraviolet light and are visible as dark patches. Here's one. Here's another. That's 3 bases in all. And the fourth is right down here at the bottom of the paper. But which is which? We've run alongside the sample 4 samples of pure bases. This is where guanine was put on. Here we put on adenine, cytosine, and then thymine. The guanine sample has reached this level. So, from our sample, the bright spot must be guanine. The adenine is the second one. The cytosine is very near adenine and takes a long time to separate from it. Thymine is the fastest and reaches very nearly the bottom of the paper. Here's the chromatography paper after it's been dried. The 4 bases, here, here, cytosine, and thymine. And I think you can see now why their position had to be marked with a pencil outline. Well, now we know what's there. The next obvious question is how much? This can be done by cutting out each spot and washing off the base. The quantity of each base present can then be estimated chemically. And if the amount of DNA in the initial sample is known, the proportions of the 4 bases in the sample can easily be calculated. When these calculations are done, we find that there seems to be a quantitative relationship between the proportions of the 4 bases. There seems to be as much guanine and adenine added together, the purines, as there is of the pyrimidines, cytosine and thymine, added together. And also, there seems to be as much adenine as thymine, and as much guanine as cytosine. These ratios were first worked out by Erwin Chargaff in 1949. He and his co-workers had examined DNA from a variety of organisms and had found that the same ratios between the bases occurred in all of them. In cows, in wheat, and in bacteria, there was always as much adenine as thymine and as much guanine as cytosine, and there was always as much purine as pyrimidine. Chargaff said of these ratios that they were striking but perhaps meaningless regularities. Well, we shall see.
Well, we've now got quite a menagerie of facts about DNA. We know where in the cell it is, we know its gross dimensions, its length and its width, and we know its chemical components and their relative amounts. But now, what about the arrangement of the nucleotides in DNA? How did people work out their arrangement relative one to the other? In other words, the primary structure. Well, knowing the overall shape of the molecule and something of its chemical properties, it's a reasonable guess, and a correct one, that DNA is composed of an unbranched chain of nucleotides, or perhaps more than one. What then of the way the chains are folded and arranged, in other words, the higher order structure of DNA? Well, a combination of 2 different approaches solved this problem. With skill and patience, DNA like this can be pulled out to give crystalline fibers. These can be bombarded with X-rays to give a characteristic diffraction pattern of spots that can be photographed. Maurice Wilkins and Rosalind Franklin used this technique to obtain this picture of DNA fibers, working in the early 1950s at King's College, London. And the pattern of spots told them something about the molecular architecture of DNA. That central pattern, the X-shaped cross, is absolutely characteristic of a helical or spiral structure. So X-ray diffraction tells us that DNA is composed of helical chains, but it doesn't tell us how many chains there are in the molecule, although in fact there had to be more than one because of the density of purified samples of DNA. Nor does it tell us the arrangement of the chains relative to each other. The answers to these problems came using a different approach, model building. And the most successful model builders were James Watson and Francis Crick, working in Cambridge. Now, they synthesized all the information available to them at that time about DNA. They knew from the X-ray data that DNA was composed of helical chains. And they also knew from X-ray data that the sugar phosphate backbone of the chains was very probably on the outside of the molecule with the bases pointing in. They knew that the molecule was of uniform width, and they also knew the detailed chemical structure of the nucleotides so that they could make up precise models of these. What they didn't know was how many chains there were in the molecule, and this is where they made an inspired guess. 2? Well, on this basis, they built their model. And it worked. Furthermore, as they built it, they realized that in order to keep the molecule of uniform width, you always had to pair a purine base with a pyrimidine base. And now, can you see another very interesting feature about the base pairing? Watson and Crick noticed that if you paired adenine with thymine, and if you always paired guanine with cytosine, this is going to tell us, in fact, this is going to explain Chargaff's striking irregularities. Because they always pair together, they must exist in one-to-one ratios. In other words, Chargaff's ratios are explained on the basis of the base pairing. But what about the all-important relationship between the structure of DNA and its function? Well, Watson and Crick were very much aware of this, and when they first published their double helical structure for DNA in 1953, they said very little about it. In fact, the last sentence of that famous paper reads, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material.